I personally believe that research is a place that can fuel hope. And I think in the last couple of decades, we've seen some pretty transformational changes in cancer treatments that really underscore that hope does exist in that cutting edge research space. I think if we want to talk about where we're going with cancer research, we have to remember what our roots are and where we've been. So I know that probably everyone on this call or listening to this will be very familiar with the backbone treatments for cancer that are still the backbone treatments today for most patients: surgery, radiation, and chemotherapy. And so what does that mean for cancer therapies? It means that we're looking at, we've really looked under the hood of the cancer cell to really understand what are the molecular changes in cancer cells, what are the targets, how does our immune system interact with cancer cells, how does the host interact, and really becoming more precise in our oncologic treatment, meaning targeting the cancer cell without targeting the normal cell, and then personalized. So your cancer may look the same on the outside, but it's very different under the hood. And so each patient may have a personalized approach to that cancer, or we may use your own cells, particularly those of the immune system, to then target the cancer.
And so when we talk about precision medicine, is the right drug for the right patient at the right time. And in fact, the NIH has actually defined what precision medicine is. The U.S. National Cancer Institute defines precision medicine as a form of medicine that uses information about a person's or a tumor's genes, proteins, and environment to prevent, diagnose, and treat disease. And the way I sort of describe it is this is what we were doing before. So if you think of weed as a tumor or a cancer, before what we would do is we just spray the whole lawn, right? Just cover it all and kill what you can. And hopefully you don't kill too much of the grass as you're doing it, but you'll get rid of all of the cancer or all of the weeds. Precision medicine is that we give a specific drug for that specific weed and hopefully leave everything else untouched.
What are we studying? Well, we now are beginning to better decipher the molecular hallmarks and drivers of cancer in the cancer cells, hereditary elements in the stroma around cancer that make it tick. And these technologies you've been hearing about are allowing us to better address these key activators that we can go after to get these treatments to our patients. So who are we talking about here? You've already heard some patient stories about real-world responses to modern therapies based on precision oncology. We're seeing such stories every day in our clinics. These are not science fiction. Mrs. SB at the top right is a patient of mine with metastatic breast cancer who was on third-line treatment, poor prognosis, and in the early days was found to have a PIK3CA mutation for which we have a targeted drug, and she's still doing very well.
I was enlightened by the transformative possibilities of modern cancer care. But sadly, it became very obvious very quickly that Canada was not embracing precision medicine when other countries were. Because comprehensive genomic profiling wasn't offered to me, I found myself forced into doing do-it-yourself cancer care in a country where delivery of world-class healthcare is expected.
The top three that came out repeatedly were: one, a lack of timely access to testing and reporting of the results to help inform those really important treatment decisions. Two, a lack of patient education and awareness of biomarker testing, due in part, yes, to a failure by clinicians to discuss the importance of biomarker testing with their patients, not because they didn't want to, but because they may not necessarily have had that time or a difficulty in understanding the subject matter and a slew of other reasons. And three, the inter and intra-provincial disparities with respect to biomarker testing access, funding, and the silos that exist within institutions and between provinces.
What really happens is if molecular testing is funded at all provincially, it's funded for a couple of genes. And so you may find a few people And then it might be available as a standard of care, and that's usually where the molecular testing is funded if there is a drug that is made available and is paid for by the government. But by and large, for many people, like Adam with rare cancer and a BRCA mutation, there is no way to access that drug.
The medical community is certainly one stakeholder of many, but there are so many aspects, so many moving parts to making precision medicine run the way it needs to run to be most effective. Clearly, governments, both on every level, provincial and federal, clearly need to be engaged. Pharmaceutical.
So what about the challenges? Well, I've hinted at a few of these. We currently in Canada have fragmented systems and various funding silos that unfortunately add a lot of complexity and time for our patients to access new therapies. Our drug pathways and our new drug funding programs are stepwise. They're often duplicative in terms of the expertise required, and they take a long time from the evidence of efficacy to access, again, over 1.5 years. We need to have programs for the pathologists and molecular pathologists to rapidly approve and have available new types of testing that they can actually apply in routine clinics or even at large community centers. We clearly need to have better access to NGS and molecular testing. We need more rapid algorithms to get those tests into practice. Right now, it varies quite a bit by institution in terms of what access they have. Do they have extra funds out of their own budgets to run these tests case by case? Is there a program? So it does tend to create a bit of a postal code lottery, even within one city, on how accessible the tests are. Patients cannot really often afford these tests out of their own pocket.
So we need to have access to molecular testing. We should have an equitable and future-proof approach to molecular testing. And when I say future-proof, I mean we don't just test the mutations that we know have targets that are approved today, which is a bit reactionary in what, frankly, is happening in most places. But we should have a future-proof concept where we test for mutations that might be important tomorrow, that we know are coming down the pipeline. And actually, most labs are already doing this. They're just not reporting the results.
What about access to targeted drugs? I mentioned that publicly funded access to drugs lags behind the evidence, often by over 1.5 years, and that will have to change. We will have to develop better models to more collaboratively and rapidly approve the evidence, like, for example, Project Orbis, looking at the regulatory end with Health Canada, and similarly, better models of collaboration internationally to let us actually approve them for regular practice and funding in Canada.
Why? What are some of the barriers? Why is NGS not something that we all do instantly, perfectly embedded in the system, 100% uptake? Cost has been a big feature, and that's been a recent challenge that we're overcoming in Canada. Time, another major issue that's in blue here. And so time, of course, if these things take too long, patients have to start chemotherapy anyway, and they miss out on their whole chance of targeted therapies.
The problem in considering these clinical trials is that sometimes these tumors are so rare to find these mutations that the patient sample is so small that we can't actually have large phase 3 randomized trials. And I think that actually has to become evident in discussions that we have when we're forming policy, is that we cannot use the same type of thinking that we use for approving other types of drugs or chemotherapies, as it were, to these targeted agents, because our clinical trials are always going to be small because we're talking about a subset of a subset of a subset. And so, when we're talking about policy and funding, there needs to be education around the people who make these decisions to understand how NGS works. Why is it that large phase 3 randomized trials are not feasible? Why is it that we're pushing for circulating tumor DNA? We need to educate people that the paradigm of cancer treatment has changed. In addition, it's not just about access. We have access to NGS testing and molecular testing for patients, but we need access to timely NGS testing. We need those results as soon as possible. And currently, we're not getting them in some situations, in some circumstances, in a timely way. And then another thing that we have to talk about is that if we're able to find these mutations, and if we're able to find these driver mutations and these light switches that are turned on, as it were, we need to have access to timely drug. So, that patient who I gave you all the way at the beginning who passed away, we don't actually have an easy way of getting that drug because it's not funded and the companies don't have a compassionate program. So it can take weeks to months to get access to these drugs, if ever, in some situations.
And that's the problem that we have today. But tomorrow, we're not going to have 30 or 40 or 50 different genes. We're going to have thousands. And we're going to have many genes in one person. We're going to have many different targets. I think it's just a matter of time. And the truth is that when most labs do test for the 1 or 2 genes that are funded, they're actually testing for many, many more. They may not be doing something called whole genome sequencing. We're really looking at the entire spectrum, but they might in fact be doing a panel of 160, 170 genes. But the reality is if only 2 of them are funded by the government, there's only really 2 that are going to be reported. And this is happening in labs all across, at least Ontario, and I think many other provinces, where the rest of the data is just sitting on the computer. It's not released, it's not used, it's not available.
And I think the prices will certainly drop. So the prices for testing have already dropped tremendously. Many of you may remember how expensive the Human Genome Project was so many years ago. And now you look at the prices of a few $100s to get comprehensive NGS panels in Canada for our cancer patients. However, the prices will probably increase for the drugs. This research is not cheap to do. The regulatory complexity around them is very expensive and contributing a lot to the prices of the drugs. So we'll have to have some framework again to really rapidly assess the drugs and their cost effectiveness to allow us to use them.